comfortable position that I have. I might have to do a floor break.
Sitting down not as comfortable.
Yeah, sitting down is not comfortable either. It's like a deep posture muscle.
Like laying down just flat.
That feels really good. But I got a terrible night's sleep last night because our bed is too soft, and no matter which position I was in, I was uncomfortable, and I do it for a few minutes, and then I wake back up and have to move again because I just couldn't. It hurt. So anyway, it's Friday Junior. Okay. Yesterday we started talking about gene regulation. The idea that we want to be able to stop a gene from being expressed quickly if we can. Gesundheit. Part of that could be because we don't want that message to be made. We don't want those proteins to do their job for whatever reason. It could also be because we want to save energy, and we want to make sure that we're not wasting energy on proteins that are not needed. Yesterday we talked about transcription, and today I want to start with post-transcription, which is our editing. So we edit the mRNA, and we talked about this earlier, the idea that we can make alternative splicing. We can make different proteins from the same piece of DNA by splicing different pieces of mRNA together. So this is not really an on-off kind of thing, turn on the gene, turn off the gene, more that we can now tailor it to the particular type of cell that we are in, even though we know that every cell has exactly the same DNA in it. We can tweak it so that it only uses a certain subset of the DNA available to get us a different message. And so remember, we talked about the idea of there's these exons we all know about now that are going to be expressed. So exons expressed. Alternative mRNA splicing or RNA splicing can expand how many different combinations you can make and how many different proteins you can make from the same piece of DNA. So going back to our cookbook recipe or our cookie recipe, and how we can have chocolate chip cookies, or we could have oatmeal raisin cookies, or we could have snickerdoodles, or we can have a variety of different cookies with the same standard cookie recipe, but then changing out little bits of the recipe to give us something a little bit different. And that's shown here on this next page. I think this does a good job of actually showing you this, where we have sections. We have a 1 and a 2 sections of exons, and those in this particular example are going to be, in both examples, are going to be expressed, as well as 5. I should have done 5 too. But then we've got another piece, a couple pieces of DNA that in one case it's a exon and it gets expressed. So here we've got 3 being expressed, and then 4 is removed. So 4 isn't there anymore. Or in this alternate scenario, we have 4 in this piece of DNA. So it's mostly the same, with that exception of the 3 being put in here. 3 being put in here and the 4 being put in the other one. So, basic cookie recipe, but maybe we don't want, maybe we want Scotchies, butterscotch, that's the word, butterscotch chips rather than chocolate chips. Okay, so 3 or 4. So you decide which you want to put in there. Okay, questions on that? Okay, so that's editing, and how we can change things up and make something different. We can also change whether or not translation. So now talking about being in the ribosome and changing that mRNA into an amino acid sequence, whether or not that happens, and how quickly it happens. You can block the initiation of transcription, so it just can't start. And that's done by proteins that will sit in that P site and block the tRNAs from coming in. And that's one way to do it. You can also, and that would be for these are for select mRNAs. So if you're talking about a particular protein that you want to talk about, you could also, and this is a little different than what we've been talking about. We've been talking about, one particular protein gets made from an mRNA. But when an egg gets fertilized, the egg actually contains all sorts of mRNAs for all sorts of jobs. And so one of the questions that sometimes students ask me is, how is it that an egg knows what to do, what to transcribe? Well, it turns out that it doesn't have to worry about that, because in the egg already are these mature mRNAs just waiting to be translated. And so once fertilization happens, there's that trigger, and then translation of all of those mRNAs happens at once. Okay. So then, all the things that need to happen to make multiple cells and multiple cell types will start going on after that sperm hits that egg. Right. mRNAs have different lifespans. Some of them are very short. They may only be a minute or two. Some of them could have hours or days or months. So you could have a mRNA that lasts for a long, or protein that lasts for a very long time, and therefore you want to make a lot of that protein. Maybe you want to make it on a continuous basis, and then maybe you don't, depending on what it is. Eukaryotic mRNA is more long-lived in general than prokaryotic DNA. If you think about prokaryotes, they're pretty short lifespan. So they don't really have to be around that long if you're going to reproduce, and if you're only going to live a few days, you're not going to have those proteins around for very long. Once a protein is made, so after you translate it, you've got this polypeptide chain, and there's modifications that will happen. Sometimes you want to take off, for example, you're going to take off that start amino acid. That methionine is going to get cleaved off. There's other modifications that can happen as well. Proteins will last for a certain period of time, and when they are ready to be destroyed, there is this chemical molecule called ubiquitin, and ubiquitin attaches to the protein. And once it's marked, that is a mark for destruction. And so there are these little guys called proteasomes, and their job is to recognize the ubiquitin on the proteins that are flagged, and then they will gobble up the proteins, and the proteins will be degraded and recycled. Their parts will be recycled. So that's another way that we can control whether or not the protein stays around and whether or not the expression has an effect. Kind of along with all that is the idea of what's called non-coding RNA. It turns out that only a small fraction of our DNA codes for proteins. Yep, there we go. Very small amount codes for that. And then there's also some of DNA that codes for RNA, specifically ribosomal RNA and transfer or tRNA. And so there was all this extra DNA, and people for a long time thought it was junk, and they would refer to it as junk DNA. And now it turns out that it's not junk DNA. It is helpful. We are just kind of touching the surface on what that function is, but we do know that it does have a function. So a significant fraction of that, quote, junk DNA will actually get transcribed into what are called non-coding RNAs, or ncRNA. And what that means is they don't code for a protein. They're not going to be expressed, but they do seem to have a role in regulating what's going on with gene expression. So I'm going to give you a couple examples of that. One is microRNAs or miRNAs. And these are RNAs that can bind to the transcript. They can bind to the mRNA transcript. And when they do that, RNA is not supposed to be double-stranded. So if they bind to the mRNA transcript, what it does in effect is now it targets them as wrong, as you're not supposed to be here, and that will target them for destruction. That will cause them to be broken down if they have that that double-stranded area. Plus, even if they are not immediately degraded, they can also block translation because they can't fit in the ribosome. They're double-stranded. The ribosomes don't know what to do with a double-stranded RNA. Biologists estimate that the expression of at least 1/2 of human genes may be regulated by microRNA. So that's kind of huge. From going from what we thought was just junk DNA that didn't have anything to now having this huge role to play in regulation of the genes. Here's my miRNA up at the top, and it's going to bind to the existing mRNA, and it can't do that if it can't be translated in that little hamburger in the ribosome if it's double-stranded. Or the other thing that can happen is sometimes it just gets degraded. It's like, no, you're not supposed to be double-stranded. Sorry, peace out. Another one that we'll mention just briefly is this guy called siRNAs. And these are similar to miRNAs in their function and also their size. And their job is to also interfere with mRNA expression. Researchers are actually using small RNAs, interfering RNAs to block transcription in certain cases, or I shouldn't say transcription, translation, to block translation so that they can see what happens. A lot of science is, well, let's see if we do this, what happens? Right? So they try something and then see what the outcome is. How does it change from what you thought it would be? You don't really have necessarily, you don't necessarily know what it's going to happen. So they can do this. The rest of this chapter is, I believe, slides about regulation. So it's just talking about the different ways that you can regulate it. This one's the overview, and then this is parts of that overview broken down into larger slides. All right. Whew. I think I'm actually going to stop there. Yeah, you're welcome.